Today, everyone, I am Vivian Tan, and together with Rohida Ali, Regina Cabiles, Erniel Ekle, and Irish Tutuhan, join us as we uncover how CRISPR is not just advancing science, but rewriting the future of life itself. We will look into one of the most important breakthroughs in modern biology, CRISPR and the future of gene editing. CRISPR changed the way scientists study genes because it allows us to edit DNA more easily, faster, and more precisely than before. So to start, I will briefly discuss how CRISPR was discovered, why it is important, its applications, and the current status of gene editing in the Philippines. So to understand CRISPR, we first need to look at where it came from. So what is CRISPR? It stands for clustered, regularly interspaced, short palindromic repeats. This wasn't something scientists invented in a lab from scratch. It was actually discovered in the late 1980s in bacterial genome. At first, they did not know its purpose. But the real aha moment came in 2002, thanks to a scientist named Francisco Mojica, when he figured out that CRISPR is basically the bacteria's own immune system. It is how they fight off viruses. They clip out a piece of the virus's DNA and store it like a wanted poster, so they can recognize and destroy the virus if it attacks again. So, right from the start, this was a cellular mechanism, nature's own gene editing tool. This is where it gets really exciting. So the big breakthrough for us in molecular biology is that CRISPR is programmable. So this was realized by two scientists, Dr. Emmanuel Charpentier and Dr. Jennifer Doudna, that this discovery earned them the Nobel Prize in 2020. So this discovery has transformed molecular biology. It provides an unprecedented ability to edit genomes with precision, speed, and efficiency that was previously unimaginable. Before CRISPR, scientists already had gene editing tools such as meganucleases, the ZFNs, and TALENs. These tools were useful, but they were more difficult to design because scientists had to engineer proteins for every target DNA sequence. CRISPR became a game changer because instead of creating a new protein each time, scientists only need to change the guide RNA. This made gene editing more accessible to many researchers worldwide. The Philippines is becoming active in the field of gene editing. Institutions such as the Philippine Genome Center support research in genomics, biotechnology, agriculture, and health. The country also has developed regulations for genetically engineered and gene-edited products. Current research includes using gene editing to address agricultural problems such as banana diseases and developing improved crops. Although the Philippines is not yet a global leader in CRISPR, it is building the foundation through research, regulation, and innovation. So, to give you the roadmap for our discussion today, at the end of our presentation, we will be able to explain the cellular and molecular mechanisms of CRISPR-Cas9, review the current advances and applications in medicine, agriculture, and biotechnology, and also discuss future directions, the ethical implications, and applications in basic education.
Thank you, Ma'am Viv. To continue, I will discuss the scientific foundations of the CRISPR technique as a gene editing tool. The CRISPR system comprises of two components, a guide RNA or the gRNA that is specific to the target DNA sequence and a CRISPR-associated endonuclease or the Cas. The Cas protein functions as a pair of molecular scissors, while the gRNA is the GPS that guides it to the appropriate site. Aside from these two, we also have the PAM or the photospacer adjacent motif. It's a short sequence of nucleotides that must be present downstream of the target site for the nucleus to make a double-strand break. These are some of the variants of the Cas9 which are mainly used for genome editing, therapeutic editing, germline and somatic editing, among others. To illustrate, this is CRISPR-Cas9 system showing the guide RNA and SP-Cas9 nucleus, which together form a ribonucleoprotein or RNP complex. The presence of a specific PAM in the genomic DNA is required for the gRNA to bind to the target sequence. The SP-Cas9 nucleus then makes a double-strand break in the DNA as denoted by the red scissors. The gRNA consists of two distinct segments of RNA. We have the CRISPR RNA and transactivating CRISPR RNA. In natural contexts, guide RNAs form a duplex molecule with the cRNA and cRNA segments annealed together. However, guide RNAs can now be synthetically produced with cRNA and truck RNA connected by a linker loop. These seamless molecules are called a single guide RNAs or sgRNAs. For many CRISPR-Cas9 experiments, Cas9 facilitates the gene editing process by creating a DSB or double-strand breaks in the DNA. Generating a DSB is the initial step in the CRISPR editing pathway. If the experimental objective is to permanently disrupt a gene so that no functional protein is made or a knockout, then the non-homologous end joining may be used. NHEJ ligates the DNA ends back together, but it is prone to error and may insert or delete nucleotides in the process, inducing a frameshift mutation. On the other hand, if the objective of the experiment is to replace the targeted genetic element with a different sequence in the case of knock-in, the cell can be directed toward the homology-directed repair or HDR pathway. Altogether, this is the summary of the CRISPR mechanism as a tool for gene editing. The components of CRISPR technique: first, second, the formation of ribonucleoprotein or RNP complex, third, Cas9-mediated double-strand break, and of course, repairs that may be done through NHEJ repair or homology-directed repair as shown in 4A and 4B. Now, to better illustrate, I have also adapted a video presentation to better illustrate the CRISPR mechanism. Scientists have long imagined being able to make specific changes to the genetic code of a cell or organism. A revolutionary genome editing technology makes this possible using an RNA-guided protein called Cas9. Cas9 searches through the cell's vast genetic material until it finds a sequence of DNA that matches the sequence of its programmable guide RNA. The guide RNA enables Cas9 to open the DNA helix, then position two molecular blades to cut each strand of the helix. Repair enzymes seal the gap in the broken DNA, sometimes by inserting new genetic information supplied by the cell or the experimenter. In this way, disease-causing mutations can be corrected by changing the underlying genetic code. Scientists are now using this Cas9 technology for research application in human health, agriculture, and bioenergy.
Greetings of peace to everyone. I am Norhida J. Ali and I will present the current advancement and application of CRISPR. So the first figure illustrates double-strand breaks DSB-based genome editing using the CRISPR-associated nucleases Cas9 and Cas12a guided by sgRNA in Cas9 or cRNA in Cas12a. In the next figure shows the base editor, a Cas9 nickase and adeninase enzyme, the prime editors, a Cas9 nickase, reverse transcriptase, and PEG RNA, the transcriptional modulators, the activated Cas9 or the dCas9, and RNA editors use Cas13 to edit messenger RNA. Now, let's discuss their applications across various fields. You know, CRISPR has transformed medicine through a wide range of applications. CRISPR nucleases, base editors, and prime editors are used to create targeted gene knockouts, knock-ins, and mutations, allowing researchers to investigate gene functions and model human diseases. Next is the applications of CRISPR, gene editing, and crop breeding. By introducing precise genetic modifications, CRISPR can improve important agronomic traits, including increased in crop yield and enhanced nutritional quality. The last one is the application of CRISPR-Cas technology in biotechnology and environmental science. So in biotechnology, CRISPR is used for the genetic engineering of crops and microorganisms. Collectively, these applications promote a sustainable agriculture, reduce environmental impacts, and help mitigate the effects of climate change. Next are the strengths, limitations, and challenges of CRISPR technology. Focusing on the strength of CRISPR technology, it enables highly precise and programmable genome editing. Second, making genome editing safer and more reliable. Third is simple programmability and adaptability allow CRISPR to be applied across diverse fields. On the other hand, we have the limitations. They are the specificity, targeting scope for the PAM, the controlling editing outcome, and the delivery. Lastly, although CRISPR has made significant advances, there are several challenges remain. These include improving the safe and efficient delivery of CRISPR components, identifying suitable gene target, and in addition, the ethical and societal implications, particularly regarding human germline editing. Thank you. That concludes my discussion.
Let us look at the future directions and societal implications of CRISPR and gene editing technology. CRISPR has evolved into a massive therapeutic platform, but moving from the lab to actual clinical trials brings major challenges. According to G et al. 2025, full therapeutic potential rests on four big hurdles and one massive technological shift. First is delivery. We need customized lipid nanoparticles or safer viral vectors that achieve transient expression, getting the editor gene to do its job and disappear before the immune system flags it. Second is balancing efficiency and precision. While advanced base and prime editors are highly precise, integrating large gene fragments efficiently still requires heavy optimization. Third is long-term safety. In vivo editing, risks of target mutations or immune rejection if the CRISPR enzyme persists too long. We must balance therapeutic durability with cellular safety. Fourth is scalability. To overcome complex, expensive manufacturing, the industry is shifting toward automated processing platforms and off-the-shelf universal donor cells. Ultimately, the most exciting frontier is the convergence of CRISPR and AI. Combining AI's predictive power with CRISPR's editing capabilities is stepping squarely into the next era of precision medicine. Moral and ethical implications. CRISPR-Cas9 allows us to actively rewrite life. Grounded in Brokowski and Adley's landmark 2019 study, highlights five critical ethical crossroads we must navigate. First is physical safety, which is a major concern, including limited on-target editing efficiency, incomplete editing, and inaccurate on or off target mutations that could result in serious injury, disability, or death to research participants and their offspring. Second is on biodiversity. It highlights the theoretical risk for damage to ecosystems and unintended ecological imbalances that could arise if gene-edited species are released into the wild. Third is equity, as expensive tools threaten to widen socioeconomic divides and blur lines into modern eugenics. Fourth is our relationship to nature. Modifying the human gene pool and altering foundational biological processes introduces unique challenges to how we interact with and change our environment. Finally, decision-making. We lack the global framework to govern these irreversible choices. As we stand at this frontier, our moral frameworks must evolve just as fast as our science. This infographic outlines the polycentric global governance framework for CRISPR technology in 2026. Without a single binding international treaty, governance relies on three primary pillars. First is the WHO framework or the World Health Organization framework, which provides ethical guidance and a mandatory registry for human genome trials. Second is the Cartagena Protocol, regulating agricultural and environmental risks. Third is the national implementation sector, where individual nations like the US, Europe, and China enforce their own domestic laws through agencies like FDA or EMA. Ultimately, these layers can interconnect to show the critical, ongoing dialogue required to navigate a fragmented legal patchwork, structural enforcement challenges, and global health inequities. Now let us see the global divide on gene editing, based on Mir et al. 2022. The table shows that gene editing laws vary widely depending on where you live. The US strictly regulates human therapeutics but leaves agriculture unregulated. Conversely, the European Union heavily restricts both, while Japan takes a middle ground. It's one technology, but three completely different legal paths. Here is another example. Here is a map showing a wide shot tracking across the globe on how does the world regulate genetically modified and gene-edited crops in 2026. This is all about regulatory orientation. Zooming in the blue-shaded countries, countries in blue like the US and Australia, focus on a product-based approach, looking at the final crop rather than how it was made. But move to Europe or Asia, and you'll find process-based or hybrid systems. This shows that the same technology is used but have entirely different global rules.
Now let us move to the final section of our presentation, which focuses on the application of CRISPR and gene editing in basic education. A common question is, why should we teach CRISPR to students? The answer is simple. CRISPR has become one of the most important breakthroughs in modern biology. Its application already extended to medicine, agriculture, environmental conservation, and scientific research. Since these technologies are becoming part of our daily lives, learners should develop a basic understanding of them. Okay? So, how can we introduce this topic in the classroom? CRISPR should only be introduced after students have already learned the fundamental concept of DNA, genes, heredity, and mutation. Once these concepts are established, students can better appreciate how gene editing works. Okay? So this slide shows that CRISPR is already integrated into the previous Philippine K-12 science curriculum. Students first learn about cells, heredity, and DNA during the junior high school years. These topics provide as a necessary foundation before introducing more advanced concepts in biotechnology. So, with the implementation of the strengthened senior high school curriculum in this school year 2026-2027, opportunities for teaching CRISPR have become even greater. The new biology electives include more advanced discussion on molecular biology, genetics, biotechnology, and emerging scientific technologies, as shown in this curriculum pathway. So these are the biology electives that I'm talking about. We have biology 1, 2, 3, and 4 that is included in the strengthened K-12 science curriculum. So CRISPR exciting, students often develop a misconception about the technology. For example, many believe that CRISPR and genetically modified organisms are exactly the same, or that CRISPR can cure every genetic disease. Others think that gene editing always produces perfect result, or that it is automatically unethical. So another is students also struggle with understanding of the molecular interaction among guide RNA, Cas9, and DNA because these processes occur at the microscopic level. So as the educator or as an educator, it is our responsibility to address this misconception through accurate explanation and meaningful classroom discussions. Okay? So to make learning engaging, several instructional strategies can be used. So we have the inquiry-based learning that encourages students to investigate the current CRISPR application. We have the project-based learning, allows learners to design biotechnology awareness, campaign poster, or infographics. Teacher may also incorporate classroom activities such as DNA paper models, CRISPR simulation, ethical debates, and case studies. So there are, in addition, there are new educational technologies such as LEDNA Toolkit, CRISPR Kit, and interactive videos that can be used in the classroom. Okay? So, promoting scientific literacy and informed decision making. So ultimately, integrating CRISPR into basic education is about developing scientifically literate citizens. So students should learn how to evaluate specific evidence, distinguish facts from misinformation, recognize ethical and societal implications, and make informed decisions regarding healthcare, agriculture, and environmental issues. Okay, so now the conclusion. I'll be reading actually the conclusion of the entire report. So for the part 1, the conclusion is that CRISPR was originally discovered as a part of bacterial immune system, but has since become one of the most powerful tools in genome editing. For the part 2, its molecular mechanism relies on guide RNA, Cas protein, DNA cleavage, and cellular repair pathways that allows precise genetic modification. For part 3, recent advances including base editing, prime editing, and improved Cas variants have expanded its application across medicine, agriculture, environmental science, and biotechnology. For the fourth conclusion, despite its enormous potential, CRISPR also presents ethical, regulatory, and societal challenges that requires responsible governance. Finally, in my part, which is the part 5, integrating CRISPR in gene editing into basic education promotes scientific literacy, critical thinking, and ethical decision-making, helping prepare learners to become scientifically informed and responsible citizens. So, we declare the AI usage as a part of responsible academic practice. We acknowledge that artificial intelligence tools, specifically ChatGPT and Gemini, were used only to generate some of the images included in this presentation. So, these are the references that we use. Now, this slide contains all the references that we use all throughout this presentation. So now that concludes our presentation on CRISPR and the future of gene editing. So thank you very much for your time and attention. We hope this presentation has provided a deeper understanding of CRISPR, its scientific foundations, its applications, and its importance in basic educations. Now we are ready to answer any question you may have. Thank you for listening and God bless.